So in the previous classes we discussed about the development of the male and female gametophytes and what is the process of fertilization, what are the changes occurs before and pre-fertilization and post-fertilization processes, and what are the main organs will produce it in the process of fertilization. You already know it, that is the fusion of male gamete with egg will result the formation of the zygote, and the secondary nuclei of the male gamete will fuses with the polar nuclei, then it will be result the formation of the endosperm. So today we shall discuss about the endosperm and endosperm morphological nature. The endosperm is a gametophytic tissue whose growth remains arrested till the entry of the pollen tube. So understood now? The endosperm is a gametophytic tissue. The growth will remains arrested till the entry of the pollen tube. When the pollen tube will enter into the embryo sac, then only the formation of the endosperm will occurs. The endosperm is the second embryo which has been modified to meet the nutritional requirements of the developing embryo. So according to Strasburger, he compared the endosperm of the gymnosperms with the endosperm of the angiosperms. So both are analogous structures being nutritive in function. So mostly the endosperm is nothing but it contains the rich nutrients like proteins, carbohydrates, sugars, etc. So according to him, the endosperm is nothing but it is analogous character in between the both angiosperms as well as the gymnosperms. In gymnosperms, it is formed before fertilization. Understood now? In gymnosperms, the endosperm is formed before the fertilization, but in angiosperms it is formed after the fertilization. In the previous class we already studied that in angiosperms how the endosperm is produced. So today we shall discuss about the development of endosperm in plants. In the previous classes we studied about the development of megasporangia and female gametophyte. So what is the fertilization, how the fertilization occurs in plants. So prior to this lesson, just you recall those points. So the endosperm is formed by the fusion of double fertilization. Generally the fertilization occurs in angiosperms by the fusion of one male gamete with the egg cell, and another male gamete is when fused with the polar nuclei, then it will be formed as a result of the endosperm. So the endosperm is formed by the formation of double fertilization and triple fusion. So let us discuss about the endosperm formation in plants. Just you see in this slide how the double fertilization is occurring. That means what is the result of the double fertilization. See here the polar nuclei will present at the central cell of the embryo sac, and their antipodal cells and synergids egg cell will be there. After the double fertilization, there is the changes occurs in the embryo sac. That is the see here the primary endosperm cell is formed at the center with the primary endosperm nucleus, and at the region of the embryo egg cell the zygote is formed. That means there is the fertilization. After fertilization, some changes will be takes place by the fusion of the male and female gametes. That is the result of the zygote and embryo. In the last classes also we discussed it. See here the double fertilization results the formation of the zygote, that is the diploid one, and the double fertilization and triple fusion will result the formation of the endosperm, that is the triploid one. So zygote is diploid and the endosperm is triploid. So let us we discuss one by one what is endosperm. So this endosperm is nothing but the it contains the nutritive tissue developed in the ovule of angiosperms as a result of double fertilization and triple fusion. We already discussed it. It is formed by the formation of the male gamete and two polar nuclei, or the fusion of the product of the secondary nucleus. So mostly in this there is the fusion of three nuclei. That means one male gamete and the two are the polar nuclei. That's why it is called as a triple fusion. That means it contains the triple nuclei. So next, what is the mostly the characteristic, this feature is seen in all the angiosperms except in the Orchidaceae, Pedostominaceae and Trapezae families. So these except these three families of the angiosperms, remaining all the families will contain the formation of the endosperm. See here the structure of the endosperm. In this slide, example like one dicot plant and another one is the monocot seed. One is the dicot seed, that is the bean seed is example for the dicot, and the monocot is for corn seed. See how the different parts will be appear in these seeds: epicotyle, hypocotyle, radicle, and seed coat, etc. It is very clear to differentiate the dicot as well as the monocot seeds. So how the development will occur? Let us see. You already know the endosperm is a result of the triple fusion. That is, the primary endosperm nucleus is formed. It divides prior to zygote and forms the endosperm. Endosperm is a nutritive tissue for the developing embryo, and endosperm is not formed in I already told you Trapezae, Podostomaceae and Orchidaceae families, and remaining all the angiosperm plants will develop the endosperm. So depending upon the persistence of endosperm, the seeds are classified as endospermic and non-endospermic seeds. Again, they are when you see this, the seeds, if the endosperm is formed, those are called as the endospermic seeds, and without the endosperm they are known as the non-endospermic seeds. So mostly in monocots and in adoines, one of the two polar nuclei fuses with the male gamete and the diploid nucleus is formed. Then it fuses with the other polar nucleus. In any case, the primary endosperm nucleus, that means the pen means the primary endosperm nucleus is formed, which undergoes repeated mitotic division, and it will be developed into a endosperm. Clear to you now development of endosperm? Next, the types of endosperm. So on the basis of the development, the endosperm is again contains the three types. One is the nuclear endosperm, second one is the cellular endosperm, third one is the halobial endosperm. So let us see this here. During the free nuclear divisions, the micropyle will be divided and it will be developed into so many number of free nuclear. That is nothing but the nuclear endosperm. And the second one is the cellular. In the cellular, the primary endosperm nucleus will be divided it will be developed into two. That means the diploid cell, and again it further undergoes the divisions. There is no formation of the nucleus, but the cells will be have just like a network like structure. So that is called as the cellular endosperm. The third one is the halobial. It is the intermediate between the both nuclear as well as the cellular endosperm. So in this next slide we see the structure and the development of the nuclear endosperm. In this nuclear endosperm, the first and all subsequent divisions of primary endosperm nucleus are followed without any wall formation. So here the primary, that means the primary endosperm nucleus will be divided. So the divisions will occur simultaneously and without any formation of the wall layer. That means no wall layers will be formed in between these nuclei. So the nuclei are freely present in the embryo sac. This type of endosperm is known as the nuclear endosperm. So next type is I told you, that is the cellular endosperm. This cellular endosperm is opposite to the nuclear endosperm. So which means it does not contains any nuclei in it, but there is the division of the cells will be occurs. Let us see here. The primary endosperm nuclei will divide and it will develops into two nuclei of the cell, and again it further undergoes the divisions and it will develops into 8 to 16, etc. So there is the no further division, there is the no formation of the nuclei, only cells will be appear. So this type of division is known as the cellular endosperm, and it formed a tissue called as the cellular tissue. So in this there is only wall formation will occurs without any nuclei. So this type of division, this type of the endosperm is known as the cellular endosperm. So the third type of the endosperm, we already know it, that is the halobial endosperm. This halobial endosperm is in between and that means the intermediate between the both the nuclear as well as the cellular endosperm. The first division of the pen, that means the primary endosperm nucleus, result in the formation of large micropellar cell and a small chalaza cell. The nucleus of the micropellar cell divides freely, but that of the chalazal cell may remain undivided. You understood now? Here there is the formation of the two cells, that is a larger cell at the micropellar region and the smaller one is at the chalazal. The micropellar cell divides freely, but without division occurs in this chalazal end. So the chalazal cell may remain undivided or divide only a few times. The endosperm so formed is known as the halobial endosperm. In this, nuclear endosperm is primitive and halobial intermediate and cellular are advanced. So here the nuclear, that means the nuclear cells will be less in number, that's why they are primitive, and the advanced one is the cellular as well as the halobial. So next, one more important feature in this endosperm formation is there is the development of osteria. That is, the different types of endosperm produce osteoidal appendages. These are the extra growth, extra tissues which are formed in endosperm. Those are known as the osteoidal cells. These osteoidal structures are meant to absorb additional nutritions requirements for the developing embryo. So mostly they are again three types: nucleus endosperm, and cellular endosperm, halobial endosperm. So when you observe this nuclear endosperm, mostly 75% of the angiosperms develops the nuclear type of endosperm, and when you see the second one, that is the cellular endosperm, 25% of the plants, angiospermic plants, contains the cellular type of endosperm, and the last one, that is the halobial type of endosperm, 19% of the plants, angiospermic plants, develops the halobial type of endosperm. So next we move to the unusual types. These unusual types means they are mostly developed from the non-uniform pattern in the cells of endosperm. Mostly they are, according to the type, they are divided into mosaic endosperm. Some we will discuss here. One is the mosaic endosperm and the second one is the ruminant endosperm. First we discuss about the mosaic endosperm. In this mosaic endosperm, in Giamese, a part of the endosperm was sugary and a part was starchy. You already know it. The endosperm will has the two types. One is the starchy part and another one is the sugary part. It mostly we observed in the Giamese. They were of two different colors. One is the red and white, which intermerged. This is called as the mosaic pattern of the endosperm. The liquid endosperm of coconut is rich in auxins, cytokinins. These are useful in tissue culture. The endosperm of maize is rich in gelatin. So these type of endosperm also has the hormones like cytokinins and auxins as well as the gelatin. So these are nothing but they are rich in nutritive structures which are very useful in the development of the endosperm. Next, second type is the ruminate. In this ruminate type of endosperm, the ruminate endosperm has an uneven or irregular surface. Example, in Annonaceae family, we observe this ruminate type of endosperm. On the basis of morphology, 7 types of ruminants have been differentiated, namely in Passiflora type and Verbenaceae, Annonaceae, Myrtaceae, Cocolobacea type and Spigelia type. So these according to the type of the morphological differences, they are called as a ruminate type of endosperm. This next in this slide you see, this is the example of ruminate type of endosperm. This slide shows the longitudinal and cross section of the ruminate endosperm. It is clear to you now? Next, main functions. What are the main functions of the endosperm? So what is its functions means mostly the endosperm is nutritive in nature. So the nutrition to the growing embryo which will supply the nutrition to the growing embryo, which contains rich fats, carbohydrates and proteins. So it is very nutritive tissue. That's why it has rich fats, carbohydrates and proteins. In some cells, the endosperm differentiates into transfer cells with extensive wall in growths. Helps in transport of metabolites from the maternal tissue into the endosperm. May persist in the seed to furnish nutrients to the developing seedlings during germination. Also regulates precise mode of embryo development, that is primitive removal of endosperm renders young embryos unable to mature even in the richest artificial medium. So this coconut, I already told you, coconut milk contains a rich nutrition. It contains the cytokinins, so it is useful in the tissue culture practices in the preparation of the media. Sorry. Types of seeds. Based on the presence and absence of endosperm, I already told you, due to the endospermic and non-endospermic seeds, due to the presence of this endosperm, they are classified into 2 types, that is the endospermic seeds and non-endospermic seeds. These endospermic seeds are nothing but they has albuminous seeds. In these albuminous seeds, endosperm is retained in the mature seeds. So it is present a entire development of the seed, and it is used in the establishment of seedlings. So this endosperm also useful in the seedlings. For this endospermic seeds, examples are cereals, coconut and castor. So next we see the non-endospermic seeds. So in non-endospermic seeds, they are also called as the non-albuminous seeds. Clear to you now? These non-endospermic seeds are also called as the non-albuminous seeds. They are mature seeds are with this non-albuminous seeds, the endosperm is seen in the young seeds only. At maturity, it will not absorb. The mature seeds are without endosperm. So the entire endosperm is consumed for the nutrition of developing embryo. Examples, beans and peas. Pea and beans, the entire endosperm we can consume. But in endospermic seeds, we can't take entire part of the seed. So it is clear to you now.